text in here, so I'll go swipe from another workshop that I did, which is not the page I sent you, but I'll put this in. All right, are we on air?
Yeah.
All right, so hello, people in the room. Thank you for coming. This is also being broadcast, so but they'll only be seeing my screen, and they probably won't hear you. So I'll repeat your questions if you have any. So a couple things. So I'm Titus. I'm a professor over in VetMed, and I'm running a lot of these workshops around this concept of data-intensive biology, which is the idea that a lot of biologists now have data, but don't necessarily know how to analyze that data. And I'm not saying that I always know how to analyze the data either, but I've been doing data analysis and computational science for something like almost, 25 years now. And so I can usually come up with some ideas at least. And so the purpose of these workshops is sort of skills training and concepts training, and then thinking about the science all in one package. So it's not just theoretical discussions of what you might do were you to have data to analyze. But it's also not just here are the commands you run. It's why are you running these commands? How might you modify them in the situation where you have different types of data? What should you be thinking about? And then what are the challenges and flaws and issues with the kind of data that we're getting? In this case, we're going to be looking only at short read Illumina sequencing. So everything we're going to talk about today applies mostly to sort of 50 to 150 base, 100 to 250 base Illumina reads, usually paired end. It's a fairly common type of data that a lot of people are getting, a lot of people have now. And we're going to be talking about how to evaluate the quality of this data from sort of a first look, and then how to trim it in such a way that you can use it safely, or how to think about trimming it so you can use it safely for downstream applications like assembly, RNA-seq quantification, and variant calling. We're not going to talk about any of those applications specifically today. We'll actually be doing de novo transcriptome assembly on Wednesday for people that are interested in building their own transcriptome from scratch and thinking about what to do with that. Today is just going to be about trimming and quality evaluation of your data. This is the first time I've really done a full 3-hour workshop devoted to quality trimming. So I put together a bunch of materials. I'm running this in parts so that I can update the materials with the flaws that I discover in them by giving the workshop. It's an applied approach to teaching. Okay, so a couple things. So one thing is please ask questions. Just raise your hand. We have an Etherpad here. There's a link to the Etherpad on the page that I sent around. Short read quality trimming and evaluation Etherpad. The value of the Etherpad is, this is where when we have commands that we have to modify, either because I messed up in the commands that I put in the web page, or because we're doing an exercise, this will be a place where we can copy and paste commands. You can also ask questions over here in the chat, and this can be a place where people who are watching remotely, or people in the room who don't want to interrupt, can ask questions either in the chat over here in the lower right, or you can put your question here, and I'll try to remember to look at this page, or Jessica will flag me in case there's a question there. Another couple things. All of this material is open, which is to say it's freely available. It will remain freely available. This web page should stay up indefinitely. It doesn't mean it's always going to be. We're not going to update it, but we're not going to take it down. And you can forward it on to other people. You can refer back to it if you want to. You can print it out. You can do whatever you want with it. But these materials will stay up and running more or less indefinitely. Ask questions. We'll take a coffee break around 10:15 or 10:30 or 10:45, depending on how things go. There's coffee in the back of this room. The remote people have to find their own coffee. And I think that's it for the welcome. Are there any questions at this point? No? Okay. How many of you have your own data sitting on not with you, but sitting somewhere? Okay, cool. So we're going to be using stock data sets, just because that way I sort of have some basic idea, know that they're going to work. But the basic commands here will work for any data set. And we're happy, we run a data therapy session on Wednesdays from 3 to 5 p.m. where you can come with your own data, and we can help you change the commands so that they'll work on your own data, and we can help you apply this to your own data. Each Wednesday, 3 to 5 in this room. So yeah. So please feel free to come to that. Jessica will send out an email about it to you now that we have your email address, and tell you how to sign up for the mailing list for future data therapy sessions. Okay. So contrary to the installation instructions, that there is one twist. I'm trying something slightly different. Rather than having everybody run their own Amazon Web Services server for this, we're actually going to be doing everything on a hosted website called MyBinder. You don't have to worry about the details about it, but what it lets us do is run these commands without having to install anything. And I'll talk through all of this as we go through it. So you won't actually need anything other than a web browser today. But everything that we're learning should be something that will be usable on real data wherever you're doing your computation. So if that's the Genome Center computers, or a server that your lab has, or a workstation, or a server that a collaborator has, the commands in general should work as long as the software is installed. All right, so let's start by clicking on Getting Started. If there's anybody that's remote and listening, if you could just put something in the Etherpad saying that the sound is okay and the screen is okay, that would be appreciated. Okay, so right, we've already covered the Etherpad link for copy paste, and then there's a chance that copy pasting into the terminal window, into the shell window that we'll be using, doesn't work on all web browsers, and we're trying to track that down. That's why we've asked you to upgrade to the latest version of Google Chrome. If it doesn't work for you, there is a workaround, and it's one of the first things we'll be checking to see if it works. Okay, so we're going to be using four packages of software today, and all of them are freely available and open source, and you can install them on whatever local computer you have. 3 of them work on Windows. All 4 of them work on Macintoshes, on Macs and Unix machines. The 4 packages are Trim-O-Matic, which does sequence trimming, FastQC, which does quality evaluation and reporting for sequences, KHMR, which is a software that helps manipulate sequences and looks at sequence content, Bowtie 2, which is a mapper, which takes reads and maps, short reads, and maps them to a reference genome or a reference transcriptome. And then I actually also have 2 custom scripts to help us plot things, to help us extract things that are in the GitHub repository. And these are only necessary if you want to run those particular scripts, but then they're going to be freely available here. If you have a local systems administrator that manages your machine, you can point them at this, and you want to use the software, you can point them at this. And then there's also a file that contains sort of the basic installation instructions. And these are things that you can point your systems administrator at and say, these are things that I want locally on my workstation, or I want it made available to me if possible. And this should give them enough this file should give them enough to install it on whatever machine you're using. Okay, good so far?
Sounds good.
Good, all right, great, okay. Oh, one last thing. This is being recorded. So it's not only being broadcast, but it's being recorded. So, and the link will be on the DIB training page, which is, which I don't think we've actually linked to from the workshop page, but this page, which is on the DIB training website. So watch on YouTube here. So you can always come back and experience the whole workshop as many times as you want. Yeah, and then the materials are here. While I'm on this page, I think I should just mention, so we do run a fair number of workshops. The de novo RNA-seq assembly is coming up. There's Python workshop next, what, Wednesday?
Yeah.
Next Wednesday. There's workshops on version control using Git, and then a variety of other workshops. And we haven't actually scheduled some of them. We haven't actually decided on the topics for some of them yet. We've also run a lot of workshops. So if you're interested with YouTube videos, if you're interested in Amazon Web Services, regular expressions, interactive data visualization, all this other stuff, RNA-seq, Jupyter Notebook, lots and lots and lots and lots of other things. In some sense, it's almost better to come talk to us and say, tell us what you're interested in, then we can point you at where we've run workshops on that. Odds are getting better and better that we have run a workshop on that. And if enough people are interested, we're happy to run it again. And generally, when we run the workshop the second or third time, we've ironed out some of the bugs and mistakes that we made the first time we ran it. So it's not a bad idea to ask us to run it again. So whatever scientific questions you have, the worst that'll happen is we'll say we don't know how to do that, or we don't feel like running a workshop on that. So yeah. Okay, so get started. Let's do a first test. So we're going to do everything in this My Binder website, and to get started with that, click on Launch My Binder. And you should, I would suggest starting in a new tab, which you can do by doing right-click, and then, can you do it with right-click? Yeah, right-click, open link in new tab. And after a couple seconds, you should get to this page. And you'll see this is called the Jupyter Notebook console. It has Jupyter up at the top. It has a list of files. You should all have 4 files. These are all independent, isolated machines from each other, so you're all working in your own workspace. And the main thing is this is an interface to a standard Unix machine, basically. So there's nothing special about what we're going to be running or how we're going to be running it. This is just a convenient way for me to get all of us on the same page. And please ask questions if you're if you get confused. So the way I recommend organizing your windows, I have a lot of opinions about this sort of thing, is bring up another window and have the window where we'll be doing things, the Jupyter window here, and then have the tutorial window here, and that way you can switch back and forth between windows easily. Copy paste. Okay. So what we're going to do to get started, in a sec. What we're going to do to get started is go to this Jupyter console and go to New and click on Terminal. And so quick show of hands for the room. How many of you have used a shell, the Unix shell before? Okay. Okay. Most, I didn't think about this in advance, but most of the data analysis that we do for bioinformatics involves big files and long computing time. We've cut down the file sizes here so that they're smaller, but sometimes it can take hours or days to run commands. Like if you're assembling a transcriptome, sometimes that can take a day or 2 or 3. So very rarely do we work locally on a laptop with software that has menus and a graphical user interface. Typically, what we do is we run these commands on a remote computer, either a computer that our department runs or our lab runs or that we've rented from Amazon or the university has. And the usual interface for that is this, which is basically a command line. And the command line here is wrapped in a web page, but basically this is a prompt that's on some computer somewhere else in the world. I don't actually know where it is. I think it's in the Pacific Northwest, but I could be wrong. And what we do is we type commands at this in order to tell this remote computer what to do. You don't have to actually figure out what those commands are in this workshop because we've provided them all in copy-paste. The goal here is to show you the process and to run you through the thinking around trimming. But these commands are basically the kinds of things I recommend running for actual data analysis as well. So, so it's sort of like, here's what they would look like were you running data analysis. We'll run some real data analysis, and then we can help show you how to modify the commands for working on your own data. So the easiest way to run commands here, I'm going to just minimize this. The easiest way to run commands here is to try and copy paste. So to copy paste, select this echo hello world, go up to edit, do copy, or you could hit Command C on a Mac or whatever it is, Control C on a Windows machine. And then switch back to the terminal window and do paste. And then hit enter. How many people were not able to paste into their window? You can't? Anybody else? You can't paste into the window? Okay. You can, but okay. So, and that's true even if you go up to the edit menu and do paste. It doesn't work? Okay. And you're running Windows? Okay, and what version of, could you send me the version of Chrome that you're running?
Yes.
Okay, just send it to me in an email. It's okay. And the same, yeah, okay. Oh, it does work?
Yes.
Okay, great. Okay, excellent. So, so we have a solution for you. It's a little bit of a workaround, and everybody else is welcome to do this as well, but copy and paste will probably work just as well. What you should do is go back to this home menu, and this is for the remote people who are listening who have trouble with copy paste, and go to New and do New Text File. And then rename this text file, click on untitled.txt and name it commands.sh. And then put the commands in this file when you copy paste. Click on save. And then in the terminal window, type /commands.sh. And what that will do is take whatever commands are in that file and run them. Did that work? Okay, you do have to save the file.
Yeah.
Yeah. Okay, and Jessica, can you? Yeah. Okay. Okay, this will not. This will generally work actually for all of the commands. And great, awesome. Okay, yeah. So the only thing you have to be careful of is you want to make sure to get rid of all the old commands before you put new ones in. So to get rid of all the commands, do Control-A and then hit backspace, and it'll delete them all, and then you can paste in the new commands. Okay. Okay. So so these commands. So what we've done with this echo hello world is we basically told our remote computer. I am not going to use the editor window. What we've basically done is we've told the remote computer to print to the screen the words hello world. It's not a particularly exciting or interesting command. We'll get to some other ones in a little bit, and I'll walk through the commands as we run them. And again, the important thing is not just the commands, but the concepts behind them. So ask whatever questions you want as they come up. But if you get confused about what exactly it is we're running, and you can always ask me to stop and break it down, or we can go through it again in the data therapy session. Okay, so let's move on to some background. Okay, so there's, I don't know how many of you know how Illumina sequencing works. I didn't find, I know there are good videos out there, and I didn't find them in time for this workshop. But the basic idea is that you have a pile of DNA on a spot on a slide that you're looking at, and you add complementary bases one at a time, and they're fluorescently colored. And you watch that spot, and as you add each fluorescent base, you record what color that spot fluoresces with. So if you add an A might be red, and if an A is the next base to be added in a sequence, and A is red, then you'll see a red flash at the particular spot on the slide that you're looking at. And so what Illumina does is it images the slide in massively parallel. Let me see. You know what? I might just. Yeah, I did have a. I'll see if I can find a video at the break. But basically, what you end up with is a movie that each time you add a base, each spot fluoresces a different color. And what the Illumina machine software does is it takes these movies that are recorded and it converts those different colors into a sequence of bases starting at each position by location on the slide. And in doing so, it faces a number of challenges. One is that the signal intensity may diminish over time as there's more contamination or more dilution in of complementary bases in each DNA spot. The signal intensity will fall off with bleaching. There may be overlaps if you're looking at small colonies of DNA that are very close to each other, and an A and a G are added right next to each other. The signal may interfere. So there's a lot of sort of situations where whether it was green or red is a matter for a statistical statement rather than a factual statement. Right? That is, it may not be able to tell that it was with 100% confidence that it was green. So what the Illumina machine does with that information is it gives you the sequence in what's called a FastQ format. And the FastQ format looks something like this. You have a text file, and the text file has a number of what are called FastQ records. And each FastQ record consists of a sequence name, which identifies where on the slide that sequence came from. It has a sequence, which is the number of bases that which are the bases that it was recorded at each spot, starting from the 5' and moving to the 3'. It has a plus, which nobody really understands why it's there. And then it has, which is just a separator, and then it has what's called a quality score. And so the quality score is the Illumina machine's estimate of how reliable its base call is. So the lower the quality score, and we're not going to talk about quality score encodings particularly, but I'll tell you what the quality scores mean. So a low quality score says this base, I read this base as an A, but I'm really uncertain as to whether it's an A, G, C, or T. So I'm telling you it's probably an A, and then I'm giving you an estimate of how likely I am to be wrong about that. And then a high quality score is the opposite. Right? A high quality score says, when I sequence a lot of DNA, 99.999% of the time, when I call this a G, it's a G. And that's because I know what the signal intensity looks like, and I see it's sharp, and it's at this position in the sequencing run, and so on and so forth. And so when we're looking at this information, we'll look at some actual FastQ files shortly. Basically, we need to understand what these quality scores mean. And I think that the key point, the reason that trimming is even a question, is that these quality scores are statistical in nature. So if you went into your FastQ file and you said, you know what, I'm going to use my software to throw away any quality score that's less than 99.999% certain. What you're really saying is, okay, well, I forgot how many nines I just said, but 1 out of 10,000 sequences will be incorrect at this base with a quality score of, let's say, Q40. If I always throw away bases that are at that have below that quality score, then 99.99% of the time, I'm actually throwing away good data. That makes sense? Because it's a statistical statement. You're basically providing a cutoff that says, what level of error am I willing to tolerate? And if you're not willing to tolerate much error, then you're going to be throwing away a lot of good data as well because of this sort of statistical statement. So here's an actual set of numbers that I calculated. If we throw away every base with a Q value of 30, then 99.9% of the data we are discarding will actually be correct. And it's even worse than that, and I'll show you why in a little bit. It's usually because you trim the read. So if you throw away everything, if you trim a read at a low quality base, you not only discard that base, but you discard all of the data after it as well, which may be quite good. Right. Okay, so the other aspect of trimming is what are you trying to do, right? So if you're trying to do, so sort of 3 things that we use Illumina sequencing for. One is resequencing analysis. And so here's my little picture of this. And resequencing analysis is, we know the reference genome, which is here, the solid bar, and we want to find the variants, which are blue. And if there's one variant in the thing that we sequenced relative to the reference genome, all of these blue mismatches will line up with each other in all of the reads. And then all of the errors in the reads otherwise will be sort of randomly located in the reads relative to where they map on the sequence. And so we can confidently call this as the variant and these as errors. So if you're doing resequencing analysis, you have to worry about having low quality bases, because if you have a lot of low quality bases in your data set for large genomes, just by chance, you're going to have some low-quality bases that all line up at one position, and if they all have the same call, and also, so it's two things, right? It's large genomes and diploidy or polyploidy. Then you're going to have multiple errors lining up just by chance at the same location, and the variant caller is going to go, oh, well, you know, the reference genome had a G here, but now I see a mixture of A's and C's in the read, so I'm going to call a variant at this location, just due to the low quality bases. So for resequencing analysis, low quality bases are more problematic than for some other situations. But there's another use case that we have for Illumina sequencing, which is counting, and this is basically RNA-seq, the RNA-seq situation where you have a reference genome or a gene set, and you want to know how many times you're seeing a particular molecule. And there what you're doing is you're taking your entire short read, and you're aligning it to your reference gene set. You're aligning it to your reference genome or your gene set, and you don't actually really care what the content of any base is, because all you want to know is where does this read match? Does it match to gene A? Does it match to gene B? Or does it not match at all? You don't care if there are errors in it. So the only reason low-quality bases affect you in things like RNA-seq is if they cause spurious matches, if they cause you to choose the wrong gene, or if they distract, basically if they distract what gene you're mapping the read to. And so for counting, there's a pretty good argument that low-quality bases on their own don't actually really matter that much. And there's one caveat to that we'll get to in a second. And then the third common thing that we use short read sequencing for is assembly. And that's where a situation where we don't have a genome or a reference transcriptome, and we want to construct one by sequencing de novo, something de novo. And this is basically how all new genomes are sequenced, right? And new transcriptomes. And so we'll talk a little bit more about this on Wednesday, but the basic idea here is it's similar to the variant calling aspect in that what the assembler will do is it will line up all of the reads and it will look for patterns. So here it might say, well, these two are suspicious, these two errors are suspicious because they line up to each other. But what it's really going to look for is a very consistent pattern of errors that are all the same and that are all lined up at the same spot. And the difference between this and variant calling is that variant calling is usually done with less sequence. You usually do variant calling with lower coverage. So you're sequencing a genome to 20 or 30x for variant calling. For assembly, you sequence it to 50 or 60x coverage. So errors will have less impact in assembly because it's lining up more sequences, and the errors are independent, occur independently. Okay, so is this sort of clear as far as the three basic use cases for short read sequencing we're covering? Okay. So the question for quality trimming is, what effect will losing real data have on the quality of your outcome versus what effect will including spurious data have on your outcome? And I'll say this again and again and again, and I've written them down here, but basically for counting and assembly, losing real data is problematic because you're decreasing your sensitivity. You're either losing low coverage regions or low abundance genes. Whereas including spurious data for counting and assembly doesn't really matter that much. For counting, spurious data won't match to anything. And for assembly, spurious data will be essentially random, and so it won't assemble into anything. Whereas for resequencing analysis, you have the opposite effect. Losing data is maybe a problem, but including spurious data is a bigger problem because there you're going to be miscalling, you may be miscalling variants in your reference. And I'm not completely making this up. I have some references at the end that I can point you towards if you want to see some of the numerical arguments. Okay, so the basic recommendations, and now that I posted this, I'm sure I'll get a lot of feedback on why I'm wrong. But the basic recommendations are always adapter trim. So we haven't talked about adapters yet, but these are the way that the sequencing machine primes its sequencing of DNA molecules. And if those show up in your data, they're never useful, right? They're never good things. They're always things that don't match your reference because they're common to your experiment or common to the sequencing process. Impose a length filter to throw away short sequences. So if you have a sequence that gets trimmed down, if you have a 100 base sequence and it gets trimmed down to like 20 bases, that's going to match a lot of places in the human genome or the human transcriptome or whatever transcriptome you're looking at just by chance, because 20 bases can match just by chance to a lot of things. So throw away those short sequences. The short sequences are going to mismatch. So you don't want to use them for counting. They're not useful for assembly, and they're generally not useful for variant calling either. And I'll show you how to do all of this. This is just sort of the upfront discussion. And then for quantification with things like RNA-seq, trim lightly. For RNA-seq, for assembly, including RNA-seq assembly, trim lightly, and I'll show you an example of lightly versus stringently. And then for variant calling, trim stringently. And here's the thing that will make you all hate me, which is that there are no hard and fast rules. So there are situations, for example, for variant calling. Suppose you're doing genome-wide association studies, right? There, depending on your linkage disequilibrium, you may care more about what variants are correlated with each other across a broad range of your sequence than any single variant. If you're looking for a single variant, you probably generally want to trim that you don't already know. You generally want to trim quite a bit. But if you're looking for a pattern, if you don't care if all you want is a 900 out of 1,000 variants that are in a particular genomic region, it doesn't matter how much you trim. You're looking more for sensitivity because you're going to identify the region based on many different variants all being in that region. And so you don't need to trim. I don't know if that makes sense, but basically there's biology behind all of this, right? And that's always the problem, is where do you make those decisions? And so part of the goal of this workshop is for you to give you some ground rules, some ways of looking at things, and basically tell you, like, look, my guess is that trimming, this probably defeats the purpose of the whole workshop, but my guess is trimming really isn't going to impact your outcome all that much. So do something that's more or less the same to all of your sequences. Look at your data to make sure it's not totally screwed up, and then get on with your analysis, because the trimming is unlikely to be the part that's really the problem. It could be, right? In like 1% of the cases, maybe, or 10% of the cases, depending on, whether you're working with dirty experiments or weird bad genomes or whatever, then your sequence could be bad quality. But in general, we're pretty good at Illumina sequencing now. So this is really more about just making sure there's not something obviously wrong with your data, picking a default trimming strategy, and then moving on with the rest of the analysis. So the difference between light and stringent trimming matters not just because you're getting rid of good bases, the good bases with the bad quality score, but also because the way trimming generally works is that you take your whole sequence and you move along the sequence 5' until you find your bad region, and then you trim at that region. And what it gives you is it gives you a trimmed sequence, but also a bunch of data from the 3' end of your sequence that's been discarded. And a single bad base here may not indict the rest of these bases, right? So generally, the challenge is that you could be throwing away 5, 10, 20% of your data by imposing stringent trimming. And that data may be perfectly good. And as of yet, with really short reads, there was never really any point in keeping this. And actually, there aren't programs that do a good job of making use of this data. But in theory, there's no reason we couldn't say, you know what, we want to ignore this base, but use the rest of these bases. It's not how things are currently done. So typically, the way trimming is usually done is this base is eliminated, and everything after this base is also thrown away. And that can actually have a really significant effect on your coverage for things like assembly, 10 or 20% loss in coverage. And I'll show you some numbers, the difference between stringent and robust trimming. Okay, so I always like throwing in some horror stories to motivate people. So this is my favorite one. It's a blog post from a colleague named Joe Pickrell. And it comes from 2012, where some people reported that they had observed over 10,000 sequence mismatches between messenger RNA and DNA from the same individuals. So RNA-DNA differences. This is typically used as a way to detect RNA editing, right? You have a genomic sequence, the RNA comes from the genomic sequence, then after transcription, the RNA is edited in some way. And this does play a role in neural development in humans and also in cephalopods. And it's interesting stuff. But it was a little bit of a surprise that there were over 10,000 sites that were discovered. And so it turns out that it's a bad sign when Science publishes an article and then like a couple weeks later publishes multiple technical comments on your article, because it means you screwed up. And what it turns out that what had happened is that they conclude that at least 90% of these RNA-DNA difference sites are technical artifacts. And the reason they figured that out is they looked at a plot here which is the position of alignments covering these sites. So they take the reads, they align them, they take the RNA-seq reads, and they align them to the genome. And then they ask, where in the alignment are there differences? And I'll show you how to make this plot at the end of today for a read data set. They said, in the read, where are the mismatches, right? So something that I didn't have a good diagram for, but in general, shotgun sequencing is selected randomly from your genome or your transcriptome. So you wouldn't expect to see at position 30 in your reads always the same kind of error, always the same kind of difference. You wouldn't expect to see systematic patterns in your reads that were real biology, because they're selected randomly from the genome. So if what you see is that most of your variants are either at the beginning or at the end of your sequence, that's a pattern that is a non-random pattern with respect to your reads, and it tells you that probably these are errors in your sequencing. These are from errors in your sequencing. So basically, what they did was they took their reads, they didn't trim them very stringently, they aligned them with the human genome, they called mismatches, and then they found that many of these mismatches were at the beginning or end of the reads, the original authors did this alignment in an uncritical way and didn't look at this pattern, didn't look for sort of known sequencing artifacts. And this is the sort of thing that more stringent trimming could have helped with, and also just looking at your data in particular ways could also help with. And again, I'll show you how to do this for a given data set at the end of today. So the other paper that I wanted to show you is from a friend and colleague, Matt McManus, and he became suspicious of the advice that you should trim RNA-seq data stringently, and he has some great figures in here that show that basically you lose a lot of information the more stringent you're trimming. And so basically, here you're losing. Let me just put it this way. So these are all the colored lines are different data sets. The Fred score here is the quality score at which they were trimmed. So to the left is more stringent, and then to the left is less stringent. Sorry, because a Fred 2 means that there's very high probability that the base is an error. A Fred 20 is a very low probability, a 1 in 100 chance that a base has an error, 1 in 1,000, 1 in 100, I forget which, that a base has an error. And if you trim at these these high quality scores, so this is less stringent trimming to the right, you actually, more stringent trimming to the right. The more stringently you trim, the fewer exons you recover in de novo assembly. So if you're taking your transcriptome and you're trimming the RNA-seq reads stringently, you actually lose out on detecting genes that are truly present in your sample. And this conclusion also works equally well for sensitivity in terms of RNA-seq expression analysis. Basically, you're throwing away data. You're throwing away a lot of good data along with the bad data. And the good data that you're throwing out affects both your recovery of transcripts and your ability to estimate the true abundance or expression level of these transcripts. So these links are in are in here, and they're worth just checking out. These are fairly common kinds of things to think about when you have sequence, and just reading through the logic of how these errors were detected and whether or not to trim is worth reading, even if you don't ever do these experiments yourself. Okay. Last but not least, we're going to be looking at FastQ files. And FastQ files get confusing basically for one kind of silly reason, which is that they really come in two formats. One format that the FastQ file comes in is interleaved, and that's where, so let me back up. So this is for paired end sequencing. So if you're doing paired end sequencing, you get from a single fragment of DNA, you get two sequences. You get one end and the other end. You can't really call them the 5' end and the 3' end because it's DNA, but so you could with RNA. But so you get two sequences from both ends of a fragment. You want to keep that information together because the two sequences from either end of a fragment, you actually know with very high probability that they come from the same original molecule of DNA. And this can be really important in situations where you're looking at splice variants, for example, where it's important that you know that these two sequences came from the same physical RNA molecule. It's also important in linkage analysis in variant calling, where you want to know that this variant and this variant, a variant called from one read and a variant called from its partner read, are actually on the same physical chromosome or same physical piece of DNA. And this information can be delivered to you in two ways. And so this is where sort of the interesting biology or methods question, which is, hey, I have a paired end read and I want to know that and so I got two sequences from the same fragment and I want to keep these sequences together, sort of hits the brutal reality of computing, which is that now we have to figure out how to encode that information in some way. And the way that information is encoded is in one of two ways. Either you get a pair of files, and you'll see for the RNA-seq data set that we're going to use, that we download from the sequence read archive, we'll get two files for the RNA-seq experiment. One is what's called the R1 file, which is the first of the sequencing reads from each fragment, and then we'll get a second file, which is the R2 file, which has the second of the sequencing reads. And in these files, there will be a bunch of records, and they'll be named something like it'll be longer, but they'll be named something like name/1 and name/2. Name/1 tells you that that's the first of the pair. Name/2 tells you it's the second of the pair. And they're going to be in different files, and they're going to be what's called in registry, which is to say the first record in the first file will be the partner of the first record in the second file. The second record in the first file will be the partner pair of the second record in the second file. And third in the first file will be the partner of the third in the second file. And you really want to keep that information together. There's sort of reasons that I've never figured out how to explain easily, but once you break up these pairs, it's really time-consuming and computationally expensive to reassociate them. So when you're processing your data, you want to keep them sort of in registry. The other way data is delivered, and the other way tools take it in, is in what's called interleaved, where you put this information all in one file. And in this file, you have record A1, record A2. So this is first sequence and its partner, second sequence and its partner, third sequence and its partner, right? And for whatever reason, the sequencer delivers files in this format, probably because that's actually, in terms of temporal time, that's what the machine actually outputs. It puts out the first sequences first, and then produces the second sequences. But then this interleaved format's actually better in many ways for computers, which are happy to look at one file and just walk through one file rather than looking at two files. And many people disagree with me on this, but I'm right. Okay, so there's a third format, which is orphans, and orphans are where you can't rely at all on this interleaved or paired format. It's just a bunch of individual sequences. And orphans are either from single-ended sequencing, where you don't have a pair, so they're orphans, or where you've discarded one or the other of a pair because it's got errors in it. And we'll show you how that works with Trimmomatic. Trimmomatic will walk through a file in this split format, and it will say, oh, well, seq-A1 is fine, but seq-A2 is low quality, so we're going to discard it. And now you end up with seq-A1 being an orphan and seq-A2 being discarded. And so these are things you have to manage when you're doing this. The simplest thing to do is often to just discard the orphans and only keep the pairs. They have more information. They're better sequences in general. But depending on what you're doing, you can also track the orphans. And I'll try and show you where we're doing this. The way you know that you have split files is if you have an R1 or an R2 and an R2. And the way you know you have interleaved is if you don't have an R1 and R2. And you can look at the files, and I'll show you how to do that in a second. Okay, so I know this is a lot, like, as always, this is a lot of like gobbledygook thrown at you. So remember, it's all being recorded. You can come chat with us afterwards. And if you do end up working with sequence, you can sort of just run the commands for the first week or two and then come back and you'll understand all of this, because you'll beat your head against much harder things You'll be thinking about it in that space. So this is sort of introducing the concepts. Okay, are there any questions or anything in the Etherpad? Okay. Okay, so we're going to start by evaluating our samples. And we're going to use this program called FastQC. And we're going to start by looking at E. coli. That's some E. coli genomic data. This is actually several years old data that I really like. Well, we'll sort of look at it. Okay, so first we have to download it. And this is going to be a bunch of complicated commands that I'll walk through while it's running. So copy and paste all of this, or put it in a command.sh file, paste it in, and then hit enter. And what you should see is it downloading the data. And so I'll explain what this means twice. The first time right now. So what it's doing is it is going to this website, the bix.ucsd.edu project single cell NBT data. This is from a Nature Biotech article on single cell sequencing. And it's using the username, single cell, and the password San Diego CA to grab the data. It's actually on the web page. If you go to the web page that I linked to, it'll tell you this is the username and password. And it's grabbing it to our remote computer. So this is the kind of thing, if you get data back from your sequencing core, from the Davis sequencing core, for example, they may give you a URL to your data along with a username and a password. And if you're working on a remote computer, this would be how you can take the data from the Genome Center server and deliver it directly to your remote server rather than going through your laptop. The reason why you want to do that is if you have 50 gigabytes of data, you probably don't want to download it to your laptop and then move it to another computer. You want to move it directly from one computer to another. Okay, you will get an error at the end. And the error at the end, it'll say something like bnzip2, IO or other error, bailing out, possible reason that follows. That error comes from something tricky that we're doing. What we're doing here is this head minus 800,000 is taking the first 800,000 lines of that E. coli file. So this file has something like 36x coverage of E. coli. So just to give you an idea of what that means, E. coli is 5 megabases. 36x coverage would be 30 times 5 is like 150. So 180 million bases. Am I wrong about that? 36x coverage. No, it's not 36x coverage. It's actually 360x coverage or something like that of E. coli. So it's actually it's a little bit crazy. It's like, anyway, let's just say it's a lot of data, and we don't want to download it all at once because it would take an hour or 2 or 3, which would basically be the entire workshop. So what we're doing is we're taking advantage of the fact that we can download the first bit of the file, and we're doing that by specifying this 800,000. This 800,000 gives us the first 800,000 rows of the text file, which are equivalent to the first 200,000 FastQ records, because each FastQ record has 4 lines. And then the computer's complaining because basically it's saying, hey, you cut me short. And that's intentional in this case. If you don't mean to get part of the file, only part of the file, and you get an error like this, that's bad. That means something happened. But in this case, this is totally expected. If you want to look at the file, we've named it E. coli pe.fq.gz. Sorry, let me increase the font size here. Sorry, the command I ran was zless, E. coli pe.fq.gz, and you hit enter, and you'll see the FastQ format. And so the FastQ format is, again, 4 lines. It's the name, and here you can see the name /1, the sequence, which is here about 100 bases, the plus, and then the quality score. And these are actually only 4 lines that are being wrapped. That's why there's a line. And then you can see that this is an interleaved file because it's the same name as the first sequence, but the second record is the same name as the first sequence, but with a /2. Okay. And then to get out of this, type Q, and you'll be back at the command prompt. Okay. So the first thing we actually want to do with this is split the data up from the interleaved into the split. And so we have a command called split paired reads. If you copy and paste that information in, you'll see something like this. what it does is it walks through that interleaved file and breaks it up into two files that are next to each other, and then we rename those files. Okay, if this all looks very confusing, totally okay, just hang on. Just make sure that you get to the end of this gzip command, and then we're gonna go back to the console. If you need help, I can send Jessica over to you, just flag me down. Okay, if you want to see the effect of everything we've done, go back to the console. If you don't know how to get to the console, let me know. It might be, when you did the new terminal, it should have created a new tab. And the console should be the thing that says home. And in the console, you'll see there's a folder called E. coli. And this is now the directory that has your data in it. And the directory that has your data in it should look like this. Should have the E. coli pe.fq.gz, E. coli r1, and E. coli r2.fq.gz. Okay, so we'll just pause for a sec to make sure everybody's on the same page.
Yeah, so
I know, I was worried something would go on.
every time I clear it, to put it in the command and then save, and then
Oh, so that doesn't, you don't, in a few seconds, that's how old it is.
Oh.
That's the one.
And then I try to, oh yeah, but I removed it.
Okay, that's fine.
So I realized that was my first mistake.
Okay, so I'm worried about that. Yeah, okay, sorry. I'm space, dot slash. And then that will give you, actually, if you run this, sorry, I think I'll put this in. If you run this, it will always go to the command.
Oh.
In your home directory, wherever you are. Okay?
Thank you. Yeah.
Sorry.
Oh, no.
Yeah.
I put it in.
No, send me an email.
Oh, got it.
All right. So I don't know how many people are following along on the Etherpad, but if you want me to wait while you finish stuff off, put something in here. Okay. Okay. So this was really just getting to the point where you have some data. We're also going to grab some yeast RNA-seq data. So now repeat what you did before, but we're going to copy and paste this in here. I'll run this and then I'll talk through it. Sorry, here. We're going to put that in the yeast directory, and we're actually going to have to get two files here. And the two files that we're getting are from, I'll actually go show you the website, are the R1 and the R2 files, and you can tell because they have the 1 and the 2 in them. You might think that there's like a much better way to figure out whether or not something's paired end than whether it has a 1 or a 2 in the file name, and the answer is sadly no. Generally, that is not do. So, so here, what we're doing is we're going and we're grabbing data from the short read archive. And this is something that So just let me, did everybody get a chance to, did that command work for everybody? Those commands work for everybody? If you run, sorry, to run the commands, if you do /notebooks commands, yeah, that's the directory. That's the directory we're in. Then that'll work. Okay. Okay, so we're just gonna wait 1 second. Let me take a quick look.
It is running. So some of the time that's true, and some of the time it doesn't matter where you use
So the question is curl. What is curl? So curl, there's two basic commands that can be used for retrieving remote data. One is wget and one is curl. And wget is says get from the web, and curl is C URL. And it's just a joke. So I use curl. Other people use wget. They have different syntax, but they do the same thing. And what they both are a way of doing what your browser would do if you clicked on a link. But instead of doing it on your laptop, it's doing it on whatever machine you're running the curl or wget command on. Yeah, no worries. No worries. We do have an intro shell workshop that we can reoffer that will help with some of that.
Awesome. Yeah,
The only real solution, I think, to learning all of this stuff, we can show you what it looks like, we can show you what it sounds like, we can talk you through it, but is to, the best way I found for researchers to learn this stuff is to have their own data and then bang their head against a wall for a couple days, come talk to us, bang their head against the wall for a couple more days, and do that for a couple weeks, and then at the end you'll look back and be like, okay, all this stupid stuff, the syntax, what to install, how to run it, that's now easy. Now it's a question of actually getting what I need to do scientifically, which is harder, but at least a better place to be. Okay, so something so this is something that I don't think we do enough of in biology. If you what you should I almost recommend the first thing you should do when you're doing a new type of approach, unless you're developing a new kind of approach. If you're just doing something that other people have done but with your own samples, is go steal their data and run the analysis you want to run on their data, because you know roughly what the answer should look like, assuming they published it. If you can't reproduce their result, that tells you something. If you can reproduce that result, that gives you confidence that you know how to do the analysis, and that you know roughly what the results should look like for that type of analysis, and now you'll be better prepared to go do your own. The challenge with this is that you have to have access to the other people's data, and in theory, at least, you have to have access to the other people's methods, and that can be challenging because people don't always put down exactly what they did. So in this case, what we're doing is we're going and grabbing this data, the SRR 453569, from the ENA EBI, which is the European Nucleotide Archive at the European Bioinformatics Institute. And typically, you'll find these accession numbers in papers, and you search for them, and you'll come to a page that looks like this. And on this page, if you scroll down, you'll have some very useful links. And the links are under here, you have FastQ files, FTP. and you'll have file 1, and in the case of paired ends, you'll have file 2. So you can see library layout paired, and then file 1, for those of you that can see at the bottom here, it's this srr 453.569_1.fastq.tz, and then file 2 is that same thing, _2.fastq.tz. And these are things that you can directly wget or curl onto your screen. So whenever I'm looking for, so I found this data set by typing yeast RNA-seq. Right? And then I just put those URLs in. Okay, I actually skimmed the paper as well, so not totally like, let's just go grab some data. And then what we're doing again is we're doing this fun thing where we're taking the first 400,000 lines in this case of the R1 and the R2 files and putting them in yeast RNA-seq R1 and yeast RNA-seq R2-fastq.gz. So if you now go back to your console, if you go back to your home, you should see E. coli and yeast, and in yeast, you'll now have yeast RNA-seq R1 and yeast RNA-seq R2. And then in E. coli, you'll have E. coli PE, which is the original file, which we could actually delete if we wanted, E. coli R1 and E. coli R2. Okay, so now that we have this data, we should do something with it. So to do something with it, go back to your terminal, and we're going to run FastQC on all 4 of these files. And so FastQC is something I've already installed, but the FastQC command is just lowercase fastqc, and then the data file to run it on. You copy and paste that, and you run it. You should see this very nice output, which says, hey, we're analyzing your data for you. And it'll run four times. And one of the reasons we cut the file size down is not just so that we could download them quickly, but so that FastQC would finish really quickly as well. All right. And so I'll just wait a minute for everybody to get to the point where they're done running that. People are good? You get the FastQC running? Yeah, okay. So this, for those of you that have advisors that are not computer savvy, this is a great opportunity to pretend that you just did a lot of work. Because if you go look at the reports, they're pretty detailed, and you can talk about how difficult it was to get this running, and give them a bunch of information, and then they'll spend a lot of time looking at it because it's like information on their sequencing that they just spend money on, and it's awesome. So let's take a look at the report. So we can look at it for E. coli first. So you'll see in the E. coli directory, if you go into the console and you look in the E. coli directory, we'll have 4 new files. Those 4 new files are E. coli R1, fastqc.html and .zip, and then fastqc.html and .zip for the R2 files. This is in the E. coli directory of film. Good? Did you? Okay, you can copy. Yeah, so after you do the paste, sometimes you have to hit enter just to make sure it sinks in, so to speak, because the paste doesn't copy. There's supposed to be a blank line after it, but it doesn't get copied. So, okay. So how do we look at these files? There's 2 ways to look at these files. One is very computer geeky, and it's what I'm going to do. The other is you can just download the zip file by clicking on it. I'll show you both, actually. If you download the zip file, so I just clicked on E. coli R1 fastqc.zip, and I click on the download, and I get a directory in the zip file, and in that directory there's this fastqc report.html. And you click on that, you'll see this report. Good so far? It's just a zip file you download and you find the HTML file in there. Okay. The other way to do it is you can actually look at it directly off the remote computer, but it's not immediately easy to do that. You have to click on the .html file, and then it will actually bring it up in an editor, which is not so useful. And to change it from an editor to a viewer, you actually change edit in the URL. You change edit to files, and you'll get the view there. So you can do either one. You can download it, or you can look at it directly on the remote website. You'll get the same result either way. So, okay. So has everybody got a FastQC report up on their screen? Yeah? Okay. So here is your first look at your data. It tells you what the file name is, it tells you what the file type is, conventional base calls, it tells you the encoding. This is not so useful right now, but there are commands that you might potentially need down the road where you need to know that it's in a particular format. Sanger Illumina 1.9 is, I believe, the latest, the most recent kind of format. Is that right, Lisa? Yeah. 1.8 is when they changed stuff between before 1.8 and after 1.8, I think. Total sequences, kind of useful, right? You have 100,000 sequences. Sequences flagged as poor quality. Sequence length is 100. GC content is 50. So this is from E. coli MD1655. The GC content of E. coli MD1655 is approximately 50%. Okay, so if you scroll down, there's a couple pretty handy plots. So the first plot, and I'm going to decrease my screen size. You should all be able to see this on your screen as well, but the first one is the per base sequence quality. And the per base sequence quality gives you box plots of the average and standard error, I think, or standard deviation, sorry, around that average based on the position within the read of that sequence. So what it's doing is this very straightforward analysis, right? It's walking through all the sequences and saying for these 100,000 sequences, by position within the read, here's what the quality scores look like. So again, Q40 is the best. Q30 is 10 times worse than Q40. Q20 is another 10 times worse. And by the time you get down to Q3, 50% of the bases are expected to be wrong. Okay, so this is not terribly great. If you didn't trim this data, I would suggest, maybe not spending too much time believing things after about 50 base pairs into your reads, because a lot of your average sequence quality drops down by a factor of 10 by position 80. Right? You're at Q30, position 80. Okay, so that's your first visualization of your data quality. One thing I want to highlight, let's look at the R2 file in tandem here. You can either download it or you can do this edit hack. If you look at this, let's see if I can switch between the two tabs. I don't know if you've noticed, but R2, which is this one, is actually worse than R1. You can see, let's do a benchmark. Where does it cross between the green to the orange? It's at about position 73 in the R2, and it crosses between the green and the orange at position 84 or so in the, I think that's, I can't exactly tell, yeah, position 84 here. And the reason for that is that the second pair in paired end sequencing is generated later in the run, so the DNA has been handled more, in effect. There's a lot more going on in the sequencing reaction. So the sequencing chemistry is basically older and more error prone at that point for the second pair. So you'll almost always find that barring massive errors of other kinds, you'll almost always find that your first sequence, your R1 sequence, is better, has more good bases than your R2 sequence. So something to keep in mind. Okay, so going back to the R1 data set, you can see the per sequence quality scores. Again, this is now the quality score distribution. So, here you can see that most of your sequences are Q38, 20,000 of your sequences are above Q38, and then you can sort of see how it falls off. here. So this is just another way of representing this basically the same data. Per base sequence content. For genomic sequencing, you generally don't expect, this tells you, I wish there was a way for me to get the entire plot on one screen. Okay, well now it's too tiny, but so this tells you whether or not you are seeing strong biases towards a particular base by a position within the reads. So if all of your if all of your reads had a G or a C in the first base, then you would have 50% GC content. For random shotgun sequencing of 50-50 GC AT content, you expect to see 25% A's, 25% G's, 25% C's, 25% T's, if I got that right. And you'll see a little bit at the beginning, and it often comes from bias and like ligation bias, in terms of what's being which of your random fragments is being ligated to your sequencing adapters. But it's pretty even. I'll show you in the RNA-seq, you'll see if there's much bigger variability here. And then by the end, you can see that the A and the T is dropping off, and the G and the C is moving up, but it's not that big a difference. And this, I believe, is caused, at least in this version of the sequencing chemistry, which is fairly old. This is a data set from 2012. Low quality Illumina sequence tended towards G's and C's at that time in the base caller, actually. Okay. So you would expect this plot to look average. GC distribution, overall sequences. This gives you the theoretical distribution for a perfect Gaussian. So basically it builds a Gaussian for what the average GC content is of your sequences, and then tells you whether or not your reads differ from that Gaussian. It's hard to make much of this difference. If this was shifted one way or another, that would indicate that some of your reads are skewed with respect to the average. That is, there are some reads that are significantly more GC rich, or there's a bunch of your reads that are significantly more AT or GC rich than would be expected from a random sampling model. And that might indicate, that would probably indicate sequencing problems of some sort. If we have time at the end, I can show you some sequencing horror stories. Per base N content, in this case, there's virtually no N's in any of your sequences. Sequence length distribution. So this is important. So this is 100 base sequencing. And the way Illumina works is if it's going to give you 100 bases, it's going to give you, every sequence is going to have 100 bases, whether or not those bases are garbage. So prior to trimming, your sequence length distribution will always be whatever length the sequencing was, whether or not those sequences are garbage. So if you ever have a question as to whether your data was trimmed or not, you can trim data in such a way that your sequences always stay the same length. You can either throw away a whole read or keep a whole read. But if the sequence is different lengths, then it's always been trimmed in some way. If some of your sequences, I'll show you when after trimming, some of the sequences will still be 100 bases in length, some will be 90 bases in length. Sequence duplication levels. This is if you have PCR duplicates, for example, in your sample. Overrepresented sequences is if there's situations where if you have oligo contamination or something else in there, you might get overrepresented sequences. Adapter content, you can see that essentially there's no significant adapter content of any of the known adapters in your data set. Kamer content is a little harder to explain, but you can think of this as overrepresented sequences. This is not particularly bad. This is probably due to repeats in the E. coli genome. It's GAA-CATC. I don't know what that is. But if you have a long list of things here, it might be something to look at and see if those represent things that might have been contaminants from how you prepared the sample. Okay, so this is a very, you look at this and it's hard to know which ones to pay attention to. It's hard to know what's normal. It's hard to know what's not normal. The two plots that, I'll say the plots that I like the most are the per base sequence quality, the per base sequence content, and then the adapter content. If you find that there's a lot of adapter content at the end of your reads, that means you have potentially have a lot of read through, where you had a small fragment and it's reading all the way through the fragment into an adapter on the other end of the fragment. And that indicates that, and importantly, this stuff should go away. You should work to get rid of this in your adaptive trimming when you do your trimming. Okay. Let's just take a quick look at the R2 one. And unfortunately, right now, there's no easy way to overlay this graph with the other one. But if you overlay this graph with the other one, you would see that this distribution is a lot fatter to the left. That is, there's a lot more intermediate poor quality scores. Best quality is out to the right. There's a lot of intermediate quality scores in the R2 data set. You can see there's more variability in the 3' end, and also a little bit more variability in the 5' end. Sequence length distribution, again, is 100. No significant adapter content. 2 different Kamer, 7 MERS that are overrepresented by position. This could just be random. Again, I don't know what to make of it. Okay, so this is for genomic DNA. This is the kind of thing that you would expect to see. I think for decent quality, there's not a lot of contamination or weird stuff going on in this data set, but the quality is not that great. So, okay. So are there any questions at this point? Yeah, sure.
The per-sequence GC content,
yes.
if you did see some severity, there are some organisms that are like 30%.
Yeah. Yeah.
So could that also just be like a thing of the organism?
Right.
Okay.
So no.
Okay.
Actually, I have a biological reason for it.
Cool.
But so in this case, what it's doing is it's doing the GC count per read, and it's giving you, so it's comparing that to the theoretical distribution based on the average GC content of all your reads.
Oh, okay.
So it's so if you had a systematic skew due to the composition of your sample, it would shift appropriately. But you can imagine a situation, and other people in the audience can tell me if I'm wrong about this, but if you had a bacterium with, say, I don't think this generally happens, but if you had a high GC plasmid and a low GC chromosome, you might see 2 bumps, right? For the chromosomal DNA and for the reads that came from the plasmid, you might see 2 bumps in this distribution. And of course, there's no way for it to know from the theoretical, to build a theoretical distribution for that without you telling that information. I would also imagine I should look at this for a human genome, for situations where you have a lot of repeats that may be very AT biased or very GC biased. You can imagine that you would see those would represent as high copy sequences that would weight the distribution one way or the other. Okay, any questions? Okay. Okay. Yep. Oh, I'm pretty sure the FastQC the E. coli data. Okay, thanks. If you could send Sean, I think it is. If you could send me an email. I think that this last command might not have gotten run, or maybe it did get run. FastQC evaluation. I thought that pretty careful. I have the GZ commands, the GZ things on the end here. Okay, well, I don't know. We'll run through it again. Okay, so what I think we should do is take a 10-minute break, bathroom break and coffee break. And then we'll come back, we'll take a look at the RNA-seq, and then we'll do some trimming. All right? Cool. I will. Yeah, I'll leave the call here. All right, we'll be back at 10:55. All right. Nope, wrong one. Entire screen. Okay, are we good? Are you looking at the YouTube? Okay. All right, so great. Okay, so I'm going to quickly look at the RNA-seq, and then we can get onto the trimming. So for the RNA-seq, we already ran the FastQC commands. Actually, let me just do a quick check. Are there any questions as to what we've done so far? All we've done is diagnostics so far, right? We haven't actually done any trimming. Okay. And are there any questions on the pad? Okay. Cool. So let's take a quick look at the RNA-seq, and I want to show you two things, and I'll see if I can think of anything else where the RNA-seq is going to be different. So on this page, I'm going to go back up to home. I'm going to click on yeast. And I'll click on the first R1. And I'll do the hack again, but you can download the zip file. So when you're looking at the yeast FastQC, the per base sequence quality is going to be whatever the per base sequence quality is. In this case, we actually get a new report because of the way the sequence names were encoded. It can actually tell you if there are biases in the where that have to do with where on the slide the Illumina sequencer was recovering the sequences from. Here you can see there's no particular hotspots of poor quality on the Illumina slide. That's what fertile sequence quality is. You have to have the right encoding and the sequence names to get that come up. The one I wanted to show you was per base sequence content. So this is something that you'll see a lot in RNA-seq data, where the first 10 to 15 bases of the RNA-seq data are quite biased, right? So, position 7 here has a lot more T's, than it has A's and C's and G's. So you could imagine a couple different reasons why this happens. One of them could be that there's just that RNA-seq, for whatever reason, is just really error prone at the beginning of the sequence. Or you can imagine that this is actually true sequence, but for whatever reason, the RNA-seq is picking out regions in the transcripts that have this base composition at the beginning. And it turns out if you map your RNA-seq reads back to the reference transcriptome and you look at these positions, these sequences are correct in general. There's not a higher incidence of errors here. What's actually happening is that the RNA-seq has a bias in where the RNA-seq reads are starting in the transcriptome. And that bias in this case, I believe, tracked it down. I have to make sure to put this link in, but I believe it comes from the cDNA, biases in the cDNA priming of the reverse transcription of the fragments, right? So in RNA-seq, you fragment the RNA and then you random prime it. And the only thing that gets sequenced is stuff that's been random primed and converted into RT into DNA. And there's bias in where that happens. So I think that's what causes this. It's either that or it's a ligation bias, but I don't offhand remember which. But it's something that you see a lot in RNA, that you almost always see in RNA-seq. So the reason you want to know that is you don't actually want to trim these off. You don't want to trim off the beginning sequences because these are legitimate sequences. You just want to be aware that your RNA-seq reads are biased as to where they're coming from in the beginning. This split, by the way, between AAT and GC comes from bias in the genome, I believe. So if we look at per sequence GC content, you see that yeast is very AT rich. And it's, the yeast transcriptome is very AT rich. And so that's why you get, 30% AT and 20% GC there. One last comment, you don't see it here, but sequence duplication levels. In RNA-seq, you will find that for high coverage RNA-seq, which this isn't because we did a subset, but in high coverage RNA-seq, for high abundance genes, you'll get many reads starting at the same point in a gene, and it will look like PCR duplicates. Make sense? So imagine that you have a beta actin. Beta actin is going to be very high coverage in your RNA-seq sample. So there's going to be a lot of reads that start at the same position in beta actin. They're going to look like they're duplicates due to PCR artifacts or whatever. And that just happens in RNA-seq due to the abundance distribution, due to the fact that you're sequencing high abundance and low abundance molecules. So. All right. Yeah, please. You might be able to infer it. So I don't know. Let me just take a quick look through. So you might be able to infer. Oh, so sorry. For the remote people, the question was, would you be able to see genomic DNA contamination in your RNA-seq with a FastQC report? And the answer is one place where I would expect to see it is if you had often the transcriptome. Often coding regions are more GC rich than the background genome, although I don't think that's true. I'm guessing that's not true in these. And so if you saw a split in your GC, if you saw 2 bumps in your GC content chart, that might be indicative of. Yeah, if you saw like there's a bunch here and there's a bunch here, but that would only be if the genomic GC content was different from the transcriptome GC content. I would not. No, I can't. I can't think of anywhere else you would see it. Yeah. I mean, it's sort of interesting because you almost expect genomic sequencing to reduce the various biases that come from RNA sequencing, but right. That's not exactly something that you would want to, I don't know. Okay, so are people sort of clear on these FastQC reports? I mean, so I should say, I mean, I feel like I do a very sporadic or partial job of explaining FastQC. There's actually a really nice tutorial video on the FastQC website that will walk you through exactly what these plots are, if you find yourself confused at any point about what they are. But the best way, I think, to understand FastQC reports is to look at a bunch of them from your data and other people's data. And if your data of the same type looks very different from somebody else's data of the same type, then either there's something different in your experimental prep, or there's something different in your sequencing. And I'll show you some pictures of some bad FastQC reports at the end. Okay, so let's do some trimming. So go back to your terminal. And okay, so let me just say, I think I've already covered all of these. Yes. Okay, so we're going to do some trimming of reads. And to trim the reads, we're going to do 2 levels of trimming. We're going to do light trimming and heavy trimming, stringent trimming. So let's start with the light trimming. I'm saying we're going to use Trimmomatic. There's a whole manual for Trimmomatic. I'll tell you what the commands we're using are, but if you want to know in detail what they are, the manual has the info. So copy and paste this, paste it in, and actually we're doing we're starting there. Okay, and what you'll see is output that looks kind of like this. You'll see. Okay, so I'll go through the command in a second, but you'll see output that should look something like this: 100,000 input read pairs, 99,580 of them of the read pairs were kept. 161 reverse of the second pairs were removed. Of the second of the pair removed, and 61 of the first of the pairs were removed. So you have 161 plus 61 orphans, plus 99,580 remaining paired reads. So let's take a closer look at this command. The top bit is changed to the appropriate directory. This is run Trimmomatic. This is run, the PE stands for run Trimmomatic in paired mode. These are the 2 input files. Trimmomatic expects split files, R1 and R2, that are in registry. So they should be, for every record in R1, there should be at the same position in the file, in the R2 file, the other pair, right? It's the Let's see. To the Trimmatic wants things in this split format. The R1 and the R2 files like that. And then you specify 4 output files. You're thinking, why are we specifying 4 output files? The first of the output files are the first of each pair. The second of the output files is orphans resulting from the second of each pair being thrown out. The third file is the second of each remaining pair. And the fourth file here, the fourth file here is orphans where the second read remains, but the first read was thrown out. Right? So you sort of have these 3 situations. Either the pair was kept, the left was thrown out, was kept, or the right was kept, or only the right was kept. And so the R1 and PE and R2 PE are where both were kept, but they're still staying split. And then the orphans are where either the right or the left were thrown out. And then you have 4 instructions to Trimmatic. The first one is one that you should always use, which is the Illumina clip. This takes off the Illumina TrueSeq adapters. The second two commands are to strip low-quality bases off the beginning and the end of sequences. So what this will do is if there are bases with quality scores of 2, which is Illumina's signal that these are just really bad bases, at either the beginning or the end, it will just trim the sequence from either end inwards until there are no more of these bad quality bases left. This is the meat of the trimming. Here, it's running a sliding window of size 4 across the sequence and looking for windows that have an average quality score of 2, which is very, very bad, right? That's essentially these bases are wrong. And in situations where all 4 bases have an average quality score of 2, it truncates the read at that point. So again, it's doing essentially this, but it's not looking for a base at Q25. It's looking or at Q2. It's looking for a run of 4 bases at Q2. And so that's why this is a very, very lenient. This is what makes this a very lenient trimming stage, because you have to have 4 really bad bases all in a row before you truncate the sequence at that point. So most sequences are going to pass this. That's why we, in fact, we had 99 point, whatever it was, 99% plus of the sequences sticking around with this trimming. So is that, yeah, okay. Let's take a quick look at the FastQ, the FastQC output. We'll do FastQC E. coli r1.pe.fq. Let's dash pe.fq. Let me copy this and put it in the Etherpad. Where's the Etherpad? Could you? Thank you. Okay, and if we take a look at this now, and this is in the E. coli directory, what you will see is that by and large, nothing's really changed. We've gotten rid of a few sequences. Let's see, which one is it? It will be the E. coli-r1-pe.fastqc.zip in HTML. And you'll see something like this. And basically, the quality scores will not have changed much at all, this distribution, because you're really only trimming off things that were truncating things that were already down in this region of the quality scores. And that was very, very little of the sequence, right? 99% of the sequence was kept. So your trimming was very lenient. So you're not going to see much of a difference here. If you did see much of a difference, it would mean that there were a bunch of sequences that were quite bad all the way through, right? The whole sequence was bad, and so it's basically throwing away the whole sequence.
quality drop off. You know, it was very, like the last 10 years.
Right. So, so the question is, can we just look at this chart and go, you know what, I really don't like the look of the last bits? And so this comes back to a little bit of the discussion that we had at the beginning before you came in, which is that these Q scores are statistical, right? So even at a Q of 20, 1 out of 100, only 1 out of 100 of these bases are actually wrong. So if you're going to throw away data where that on average is 99% correct, that's actually throwing away a lot of data. So it really depends on what your downstream goal is. If your downstream goal is variant calling, I agree with you. Truncate once you get past below a certain point. If, on the other hand, your goal is to do assembly or quantification, then then most of this data is correct and will probably help your assembly and your quantification. So that's why it matters what your downstream goal is. But let's take a look at what happens. We can do this much more stringently. Oh, so before I want to do that, I want to show you one other thing, which is sequence length distribution. So we basically didn't change the sequence length distribution at all. But you can see that before, I don't know if you remember, this was just a pyramid. All of the sequences had a length of 100, and none of the sequences had anything else. Now there's actually sequences that have been shortened all the way down to 48. There's only 1 or 2 of them because we trimmed so few sequences, but there are sequences that are shorter. So this graph now looks very different from the original graph. So now you know that this data has been trimmed. Okay, so let's do a more stringent trimming. If we take this, and I'll show you where the command differs in a second. If we do this and we run it, now you'll see that only 88% of your data is surviving this more stringent trimming. Okay? So now you're discarding 10% of your sequence. If you look at the FastQC report, I named it the same thing, so you can actually just reload this or re-download it. If you look at the FastQC report, did I actually rerun FastQC? I didn't rerun FastQC, so you need to rerun FastQC. So FastQC, maybe FastQC E. coli R1.pe.fq. And you come over here and you reload this. Now you see a very, very different report, right? So what did we actually do? Has everybody, I'm going to wait a minute to make sure that everybody is happy with, gets to where they want to be. Is everybody needs to a new FastQC report? You guys good? Okay. So what did we actually do? The difference, the only thing that was changed in the command was this sliding window. The sliding window before was sliding window of 4 with a quality score of 2, and this is now a sliding window of 4 with a quality score of 20. So now what it's doing is it's running a 4-base window across the sequence, and as soon as the average quality score across that 4-base window drops below a Q of 20, it truncates. And that's why when you look at this, you can see that there's basically nothing that's bad. Why would on average everything improve so much, right? We're not seeing anything that even approaches 20 in terms of standard deviation, much less the really miserable drop that we saw before. So what can explain, anybody have any ideas as to what can explain this? Right? If we're just getting rid of bases that are below, if we were just getting rid of bases that were below quality score of 20, I would still expect to see a bunch of more sequence down here. Let's go look at the distribution here. So why is it that there are no sequences that approach a quality score of 20? So this is this fun thing about Illumina. This is more true of the older Illumina data, but you might still see it in your newer data, which is that a lot of Illumina sequence tends to be pretty good, and the Illumina sequence that's not good is really bad. And so as soon as you apply a fairly stringent filter, you actually get rid of a lot of this sequence that's dragging down your average qualities. And so essentially, you're not seeing many sequences that have a few bad bases that are getting truncated, but are mostly good. It's mostly a situation where you're throwing away a lot of the bad data, a lot of the bad sequences with that one filtering step. And so, just to show you what the consequence of all this is, I've already said you only have 80% of the data. You know, 88% of the data, but if you copy and paste this read stats command, you'll see, let's see if it comes up. There, if you run read stats on the input data, there's 20 million bases in 200,000 sequences of average length 100 prior to your trimming. Do you want me to come back and help? Okay. So prior to trimming, you have 20 million bases in 200,000 sequences. After trimming, you have 17 million bases, 17.2 bases with a 92.7 base average length, right? So basically, you've not only discarded about 10,000 sequences. Is that right? Yes. You've discarded over about 13,000 sequences, 14,000 sequences. You've also dropped 8 bases on average off of the sequence, off of each sequence. And so this is the difference between the stringent trimming and the lenient trimming. The lenient trimming, basically, we got rid of 1% of the sequences. Here, we're getting rid of 10% or more of the sequences. And if you spent a lot of money on your sequencing, then you have a lot of data, and it's okay to get rid of 10%. If you're sort of pushing the edges of your, if you have relatively low sensitivity as it is, then getting rid of this extra data may be problematic. Okay, let's wait a sec to make sure everybody catches up. Okay, so just to repeat here, we got rid of over 10% of our data, which is quite a bit. Okay. So what I think we might do, what I wanted to do, rather than give you the commands to copy paste, I thought we could try building our own commands to trim the yeast RNA-seq data. But let's just, since we don't have a huge amount of time, I'm going to do it a little bit collaboratively. So I'm going to start. So this is, I should say, so this is the secret to how almost all bioinformatics is done. What you do is you find a command that somebody else wrote that works, and then you copy the whole command like this. Okay. I'm going to do it in an editor. So I'm going to come over here. I'm going to say new text file. And we're going to name it rnaseq.sh. I'm going to, oh, I changed the URL, sorry, not the name. We're going to change it here to rnaseq.sh, or yeast, whatever. Okay, so can somebody, what's the first thing about, so the goal here is I have this command, this set of commands which worked for E. coli, for trimming E. coli, and now I want to change it to work to trim the yeast RNA-seq data. What's the first thing I should change? Anybody? So which? CD yeast, exactly. Okay, that was a gimme. What's the next thing I should change? So line 5 is the input. I'll copy it. I'll put this command in the Etherpad when I'm done. So line 5 are the input files. So what are our yeast RNA-seq input files named? I don't actually remember because I have the attention span of a gnat. So what I'm going to do is I'm going to go into the yeast directory. Okay, so it's yeast-RNA-seq-1. You know what? I really do have the attention span of a gnat, so I'm just going to copy that, and I'll put it in twice. And then I just have to remember to change the second one. So always the trick with copying and pasting, and this will happen to you basically all the time. This still happens to me. So you copy and paste, and you'll forget to change things. So you'll have like yeast-RNA-seq R1 and yeast-RNA-seq R1 in the line. And most programs, many programs will complain if you do something obvious like that. It'll be like, hey, you told me it was paired data, but you're giving me 2 files that are both singleton, and some won't. And you know, it's one of those situations where the reason we look at our data and the reason we look at the FastQC reports is partly because we should expect those reports to be different for different data sets. And if they're the same, then probably somewhere up above you copied and pasted something that, anyway. Okay, and then let's just change the output. So I'm just going to be lazy. I'm going to just change yeast, E. coli to yeast everywhere. Another thing that I kind of want to point out, you're going to be tempted to take shortcuts with file names because it's annoying to type out these long file names. I'm here to tell you to not take shortcuts with your file names because at the moment we can kind of predict what each one of these files is. It's even better if you did, you know, yeast trim, yeast trim, yeast trim. There's techniques like tab completion that will help you match these file names. What you really want, this is actually my my wife's favorite comment. What you really want is to be in a situation where 6 months down the road, when you've been back in the lab doing a bunch of stuff and now you're coming back to do your sequence analysis, you know what each of the files sitting in your directory is when you look at them. If you have to email somebody to figure out what they are, I will tell you that you of 6 months ago does not respond to email, right? So you want to have really clear, excruciatingly long and detailed file names so that you can look at them and go, okay, you know, Lisa trimmed this file on February 15th with these parameters. You know, maybe not quite that long, but something that's like really, really clubs you over the head with your file. Okay. Do we need to change anything else? Okay. Can anybody tell me if this is stringent or lenient trimming? Lenient. And how do we know that? Because on line 9, we have 4 and 2, not 4 and 20. Okay. You're also going to be really tempted at some point, and it may not be you, maybe somebody working with you, to just sort of go through here and go, well, I don't really like the number 50. I want to change the number to 55 because it's Thursday. People seem to do this a lot. Like it's the equivalent of putting your own stamp on your experiments. I really wouldn't bother. Just pick some numbers that you're going to use for all your experiments. Use them. Don't think about them until you have a reason to change them. Right? It's changing things sort of semi-randomly in bioinformatics is a really big problem because you never know, like, oh, did I change the quality trimming parameters for like the 3rd batch of RNA-seq, or did I remember to do it for all of them? And, you know, at the end of the day, that kind of uncertainty is not, you know, which pH buffer did I use for that? Like you've all been there, right? So just be boring is what I'm saying. Okay, so this is now the command we're going to run. And I'm going to save this. And interestingly, I created this file by accident in the E. coli directory, even though it's for trimming the RNA-seq, the yeast RNA-seq. So let me see if I don't remember how to move it. I'm going to go here. I'm going to go into E. coli, and I'm going to take the RNA-seq.sh and rename. No. Okay, I have no idea what to do about this, so I'm just going to have to use it from this directory. Okay, so I go back here, and I'm in the E. coli directory. I'm going to go into the yeast directory, and then I'm going to run. Yeah, I can move it from this family. Fine. Be that way. Okay, so what I just did there was I moved the file from the previous, from the E. coli folder, neighboring folder, to the current directory, which is what the dot did. I'm going to do dot rnaseq.sh. Actually, you know what I'm going to do first? Now it's been moved, so I have to go find it. What I'm going to do is I'm actually going to, actually I still don't have my Etherpad up. Okay, you put it in Etherpad, thank you. Okay. So, what just happened? No, that's right. So, and now I'm going to run it. You can run the command by copying and pasting it, or putting it in the command sheet, or whatever. So, it's not actually worth. Okay, so one comment. This was lenient trimming, right? But notice something. You got rid of 6% of the pairs, but 6% of the forward reads survived. So what happened there? What happened is that for whatever reason, and we can go try and figure this out, for whatever reason, 6,000, most of the R1 sequences were good, but there were a bunch of the reverse sequences that were bad, and so it trimmed a lot of those. It trimmed about 6% of the reverse sequences off, leaving a bunch of orphaned forward sequences. Okay? Just to make clear, what we're looking at is the difference here. Forward only surviving 6,000, both surviving 93,000 out of 100,000. So can anybody tell me what, give me two, there are at least two competing hypotheses for why you would drop a bunch of the R2. Any ideas? Okay, there's one, the first one that comes to mind. Anybody? The R2s are just crap here. So even with lenient trimming, it's just like get rid of these sequences. Okay. And then what's the other theory? Is there another theory? Actually, now I'm wondering. I was thinking if the reverses if they were short fragments, but if they were short fragments, then you'd expect both forward and reverse to be trimmed. If there was read through. So yeah, I think it's just got to be that it's bad sequence quality. Yeah. Okay, so I was trying to be clever, but the answer is probably just that the R2 sequence. There were a bunch of really cruddy R2 sequences. So just to take a quick look and see if that was the case, we have these FastQs. Let's go take a look at the yeast R2 FastQ. And wow, okay. And we never actually looked at the quality scores of the R2 yeast data, but that's pretty bad, right? So there's a bunch of data there's a bunch of data in the second pair of the yeast RNA-seq that's just got really bad quality scores. And so that's what we're trimming off with that trimmomatic command. All right, any questions at this point? Okay, so in terms of sort of basic trimming, that's sort of what I recommend for the basic trimming. You have your choice of stringent or lenient. You look at the data with FastQC before and after trimming. You try and understand how much you're losing and why you're losing it, and then you're done. And then you move on to your next exciting adventure. One thing I wanted to show you, I think this is a good time to show you. Just want to show you what some really bad data looks like. So about a year ago now, I asked this question on Twitter. Let's see if I can actually get the original tweet up. That did not work. Wow. Huh. It really didn't work. Let's see if that works. Nope. Okay. Well, anyway, what is this? Did I forget to do that? And is there something else I need to put in? Then status. Okay. So right, could anyone loan me some bad FastQC reports? Okay, and there are actually some, you post something like this on Twitter and you get some pretty normal responses, some pretty entertaining responses. One of them was where, I wonder if it's in here. There was the one like, there was the response that was like, what, like your data's too good? Yours are too perfect? Anyway, so, okay. So then, and then there's like the trolls. This is actually a friend. What's a bad FastQC report? Anyway, so I got some really interesting ones, and I included the link to this in the docs at the end. So sequencing bloopers, technical sequence. I just want to make sure you recognize, like, the FastQC reports we've been looking at. There's like, that's like real data. That's good. There's like good data in those files. So this is what happens when like the instrument breaks in cycle 26. Like, all right, we're done now. We're done producing good sequence now. If you see something like that, you go to the sequencing facility and you say, I'd like you to rerun my run for me. Thank you, right? Because you guys screwed up. Specific cycles lost. So you'll sometimes see situations where it's just like, for whatever reason, insufficient reagents, machine not warmed up, whatever, like barf, right? No priming signal, right? So, on the second side, there's no data in your reads, right? Like it literally isn't calling any bases, right? And so, you're toast. If you remember that tile, quality per tile, if the DNA concentrations are too high that they put into the machine, there will be many overlapping clusters, and you'll get a lot of bad quality calls in various places, and that's a sign of too much DNA. And that's actually the sequencing facility's problem, because they're measuring the DNA and deciding how much to put in. Consistent tile fail, if there's portions of the slide that are just whatever, broken for whatever reason, you'll see these sort of artifacts in the tile. Transient tile fail, incorrect Phred scores. So this apparently is a big problem. Illumina changed its encoding of quality scores. When you upload your data to the sequence read archive, you have to tell it which version of Illumina you used, and if you get that wrong, actually it turns out, this is a problem with the sequence read archive. They don't properly deal with this. So you actually get different quality scores depending on, when you download the data, it'll give you different quality scores on the beginning part of the data and the second part of the data, which is not good. Wrong barcode annotation. That's if you're doing multiplex sequencing. Contaminated barcode stocks. So I think this is just like, right, there's like nothing there. Let's see if I can find odd sequence composition. Okay, so this is what happens. You remember that adapter plot? If as you're reading through the sequence, you encounter the adapter, that's a read through because you had a very short fragment. So you read from the left all the way through the fragment to the right adapter. And you can see that the Illumina universal adapter, the frequency of the Illumina universal adapter climbs to about 100% by the time you're 26 bases into your sequence. Again, this is not your fault. This is the fault of whoever, generally. It's the fault of whoever did the fragmentation. So these are all situations where you go back to the sequencing facility and ask for more data. Adapter dimer overload. So this is if you put in too many adapters, I guess. And you just get crud. This is that bias sequence content across all bases. You can see basically this is bisulfide sequencing. You can see the incidence of Cs is very, very low. And that's because I think because you're converting all Cs to something else. So this is expected. This is from certain kinds of sequencing where you're sequencing, like you're doing targeted sequencing. You get consistent biases by position because you're sequencing a GC-rich gene or an AT-rich gene. This is the unavoidable RNA-seq one, positional sequence bias that we talked about before. Unexpected doubled adapters. I don't know what happened here, but it looks pretty ugly. And maybe two adapters got ligated at the front end. And so you have extreme sequence bias right at the beginning, right? Again, probably not your fault. Probably the sequencing facility's fault. And there's all sorts of other fun things. So contaminated with guessable sequence, right? So sometimes if you find that a lot of your data doesn't look like it's mapping to the organism that you thought you'd sequenced, you can go see if you sequenced the technician by accident. So this happened in one data set that we have access to. We're pretty sure that there was a significant amount of RNA contamination from the technician. And while we never have gone and done it, we can even identify exactly which technician it was because, of course, we now have their genomic sequence. Anyway, so there's all sorts of fun things. You know, no hits, right? It doesn't hit to human, mouse, rat, E. coli, yeast, Phaeax, adapters, vectors, wasp, which I guess was probably what they were trying to sequence. Ribosomal RNA, it's just no hits at all. So congratulations, you sequenced something new, but you don't know what. At this point, I've actually know people who have gone and then assembled this to try to figure out what it was they sequenced. So you can have fun. Let's see what else. Reagent contamination. So it turns out that that there's called kitomics. If you're doing metagenomics and you sequence something, DNA that was extracted using the kit, you also need to sequence the blank water from the kit because it has microbial contamination in it. So I have friends who talk about this a lot. Anyway. So, okay, so what I want to spend the last like 15, 20 minutes on, by the way, I should say this all comes from a 2-week workshop that we run in Michigan. And I'm happy to point you with these links. This has a bunch of tutorials on BLAST, Amazon Web Services, mapping and assembly, genome assembly, and so on. And it's in the same style as these tutorials. So it's got a bunch of tutorials. If there's one thing we have, it's tutorials. Whether or not they all work is a whole different question, but we have them. So if you're whatever you're interested in doing, we could probably find you a tutorial on. Okay. So this is really the end of the trimming bit. And I wanted to show you my other favorite trick. So at this point, you've got your sequence, it's trimmed in some way, you've looked at the FastQC reports, you know what your sort of error profile is, and now you have to decide, are you going to do the mapping, are you going to do the RNA-seq, are you going to do, mapping or quantification, variant calling or quantification or assembly. And again, there are other things you have to think about. But at this point, you've got your trimmed sequence. But there's one more thing that I would suggest you evaluate. So the big flaw, okay, I'm not a very trusting person. The big flaw with these quality values is that they come from the Illumina sequencer themselves. So if there's biases or weird things going on with a sequencer, the quality scores may not completely reflect what's actually going on. So there's another approach you can take, and it's usually fairly quick to take. And what you can do is you can evaluate the read mismatch statistics. So if you remember at the beginning when I was talking about the RNA editing miscall, I showed you that graph where like all the mismatches were at the beginning or the end of the reads. And I told you I'd show you how to generate that data, that result. That's what we're going to do here. What we're going to do is look at the error bias in read data sets, not with the Phred score, but doing something empirically, where you're actually looking at the sequence and comparing it to what you know it should be. You can only do this in situations, it's not quite true. In this situation, you need to have a reference in the first one of these two. You need to know, I sequenced yeast, so I should be able to map to the yeast transcriptome, or I sequenced E. coli, I should be able to map to the E. coli genome. And you can do it even for resequencing analysis, because for resequencing analysis, there should be no position-specific signal from the variants with respect to the reads. That is, the variants, the true variants in the genome will be located randomly throughout the reads. So if you see a signal here, then you know that it's due to errors. So let me just show you an example of how this works. We'll go back through these commands in a second if I want to run them. So copy these commands and paste them. Okay, and while that's running, what we're doing is we're going into the E. coli, data set directory. We're downloading the reference genome here. This is the E. coli MG1655 reference genome. You can get it from NCBI. I just happen to have a copy somewhere else. We're uncompressing it. We're building a mapping index called a bowtie index using bowtie build. And then we're using bowtie2, which is a mapper, to take each read and place it against that reference genome. And that produces what's called a SAM file, which if you do variant calling, you'll become very familiar with. And the SAM file basically contains a summary of where the reads have mapped to on the reference. I can draw this out on a diagram if people are unclear on what mapping is, but it's basically where do these reads match. Okay. And once that's done, which shouldn't take very long, really, it shouldn't take very long. Okay. Well, apparently it takes longer than I thought. Okay, so you get this report from the bowtie. For bowtie, it says, I'm only mapping unpaired reads, 96.6% aligned exactly once. This is the E. coli genome. It's not very repetitive. So reads randomly sampled from the E. coli genome will map once. 3% align more than one time. This is probably due to repetitive elements in the E. coli genome. And then about 0.5% align 0 times. And the reason you might get reads that align 0 times is if they're highly erroneous reads, in this case, or if they're, or contamination, because we're only aligning against the E. coli genome. Or you can imagine a situation, this is probably not the case here. Well, actually in this case, those reads could either be things that mapped over the circular junction, because we didn't circularize the genome before mapping to it, and this is in bacterium, so circular. Or so contamination errors, or things that aren't in the reference sequence. But these are very, very, this is a 99% overall alignment rate is very, very, very good. So what we can now do is we can take these 96,000 reads that aligned exactly one time, and we can run this little custom script that I wrote that takes the SAM file and walks through it and asks where in the reads by position do there tend to be mismatches. And this script outputs two interesting statements. One is it outputs an estimate of the error rate. So the error rate estimate here is assuming that the reads came from this genome, how frequently by base does the read mismatch with the genome? So an error rate of 1% or less is pretty typical for Illumina. Right? So that means 1 in 100 bases is wrong. And then there's this log ratio of forward to reverse. This just asks, is it unbalanced? Do you tend to get forward mapping or reverse complement mapping? If you're doing, what's the word? Direction-specific, single-stranded RNA sequencing, for example, well, it's not necessarily true. Yeah, if you're doing single-stranded RNA sequencing and mapping to a reference transcriptome, you would get a very strong bias. A bias of 0 basically says it's approximately 0 basically says half the reads map to the forwards to the top strand and half the reads map to the bottom strand of the E. coli genome. And what this does is it produces a file called ecoli-mapped.rhist. And if you look at that file with less, you can also download it and open it in Excel. You'll see a file that looks like this. It's a little hard to see. So could you put that command in the, you could have less E. coli map. There it is. Yep. You'll see what it gives you is essentially a spreadsheet of position within the read, the number of mismatches, they're called errors, at that position in each read. In how many reads have a mismatch to the reference at that position, and the overall fraction of location of reads that map to the overall fraction of reads that had a mismatch at that position. And so if you look at this, you think, okay, well, it's not too bad, right? Like, but what you start to notice if you scroll down by hitting enter is that the numbers go from 200 to 300 to 400 to 500 to 600 to 700 to 800, to now 1600, 1700. So what you're seeing is that the error rate at the 3' end of the reads, as you get into the read, the number of mismatches increases dramatically. To the point where you went from having about 200 mismatches at position 1 to 4,000 mismatches at position 98. And so this is an empirical statement about about the frequency of errors with respect to position within the read, the frequency of mismatches. And again, the dominant explanation, by far the dominant explanation for this is that barring other kinds of bias, is that these are errors that are occurring from the increasing rate of error as you sequence towards the 3' end of a read. Okay. Does that make some sense? You can use tail to look at the end, and you see that we're actually getting, at the end of the read, we're getting to an error rate of about 5%, where 4.6% of the bases at position 99 have a mismatch with respect to the reference. And again, the explanation for this mismatch rate is errors. This is different from the quality score, because this is no longer a statistical statement. We can actually go in, this is a summary of where the reads actually mismatch with the reference. So we're not saying like, oh, approximately 4.6 of the bases will probably not have errors in them. We're saying no, 4.6% actually have mismatches, and those are probably errors in this case because what was sequenced was this genome. If you were doing, if you're sequencing an evolved strain or a different strain than the reference strain, then you would expect some of those errors to be, some of those mismatches to be real variation. But you wouldn't expect that variation to be biased by position within the read. So I'm going to show you something. You don't need to follow along. I'm going to do the equivalent of downloading this to Excel. Let me see. Was I doing this for yeast or was I doing this for E. coli? E. coli, right? If you go into the E. coli data directory, I'm going to actually do a graph. I'm going to use a notebook, a Jupyter notebook to do a graph. And you don't have to follow along, but I'll copy and paste this. This is now Python code or the Python programming language, we're actually loading the data in and graphing it. We have other workshops on this type of stuff. And then let's do map error is equal to num_key.load text. What was the file called? I'm telling you, I have a E. coli-maps.airhist.
Yeah.
Airhist and plot. Oh, and I have to do skip rows equals 1 to get rid of the header. Plot map error. And so this is actually, sorry, so this is what the mismatch profile looks like by the position in the read and number of mismatches at that position. And so you can see there's a lot higher incidence of mismatches at the 3' end of the read than at the 5' end of the read. What true variance would do is they would raise, or what true variation would do is they would raise this. You'd have more mismatches on average across all of the positions due to real mismatches between what you were sequencing and the reference genome that you're mapping to. But the bias here is almost entirely due to errors. You typically do see things like this peak, and this peak is something that I don't know how to explain, but it seems to be generated by the sequencer in some way. Either some reagent or pump malfunction or something, or a technician. My suspicion is always that a technician walked by and kicked the machine. But okay. So this is actually a really useful graph to show yourself, to convince yourself that there's not something really weird going on. So if you see a massive spike at position 20, and you're doing variant calling, you want to make sure that whatever is at position 20 isn't, I still don't know what to do about those reads, but what I would almost suggest doing if you have a massive spike at one particular position is getting rid of all of the reads that have a mismatch at that position, because they're unlikely to help you. They're not going to help you in anything that you do down. That's an example of a systematic bias as opposed to a random error, right? To explain that a little bit more, generally in sequencing, all of the tools assume that you're doing a fairly random sampling, and anything systematic error, systematic bias are going to give you non-random results in your downstream pipeline. So you sort of want to get rid of them. I don't have a script to do that, but again, if you come to data therapy, we can help you if you run into this problem. I'm not going to show you this for the RNA-seq, although you can also calculate this for the RNA-seq. RNA-seq is where you actually will see some pretty big nasty biases. This actually is a pretty good excuse for now trimming sequences, right? At this point, I could say, look, well, is that true? I mean, we're only getting a 5% error rate at the very end, right? That's 1 in 20 sequences are erroneous at this position. It'd be pretty interesting. Let me do a quick look. So this was R1. Does this work? Let's take a look at so on average, you can't really compare these, but on average, we're saying that 5% of the sequences have mismatches at this position. So I guess what we'd have to do is sort of spin things out into quartiles or something to figure out whether or not this quality score estimation was correct or not, right? You expect this profile to follow the other profile pretty well if Illumina knew what they were talking about. But probably too much right now. So the other thing, so does everybody sort of get what we're doing here with the mismatches and why it's interesting? Are there any questions of this? So this is an empirical statement about where your reads are not matching your reference with respect to position within the read, generally from sequencing error. The only problem with this is that you need a reference. You don't have a reference sequence. If you're sequencing something new, then you can't look at it this way. Although you could assemble a reference and then look at it this way, but if the assembled reference is biased by the errors in your reads, then that causes trouble. So another thing that you can do, and this, I'm not going to go through these slides, but this is something that is related to some of the research, some of our research in my lab, is you can generate a de novo read mismatch profile. And what the de novo read mismatch profile does is it looks for, I'm trying to think of where, it looks for biases in a situation where you don't know what the genome is. It looks for systematic patterns of X's with respect to their position in the read, which is to say, you can look at these overlaps and you can assign errors based on where the reads mismatch with other reads. So you don't need a reference genome to do that. You can do it off of what's called the assembly graph. And you can do that for both RNA, for genomics and RNA-seq and metagenome data. And this is an example for RNA, or sorry, for the genome data. And the way it works is you build a database of all of the sequences, and then you build this error list from the de novo from that database. And this is a different kind of, this is what's called a Kamer error hist, but it gives you similar but not quite the same information. And you'll see instantly what I mean. It all works. So here, you can see it actually underestimates the error in the 3' end. It has some weird bits here. It's actually a little bit weird. I think this is from a bug that I actually fixed that isn't installed into the software. The main thing to note is that you actually see similar patterns of high error rates in these two profiles. And this is the kind of thing you want to look at and go, oh, I kind of want to get rid of reads that have that level of mismatch at that position. Because those reads, if I use reads that have this position to call variants, then that's a higher error rate than I would normally expect. So I didn't realize I still have this bug in the released version of PageMark. Anyway, okay, so there's some things there's more things you can do to get rid of errors. I generally recommend mostly doing the quality score trimming. There's Kamer-based trimming. That you can look at this slide if you're interested. It turns out that quality score trimming doesn't get rid of most of the errors, whereas Kamer-based trimming does. On the flip side, Kamer-based trimming will do horrible things in the face of variant calling. So it's not useful for variant calling, it's mostly useful for assembly. Don't worry about it, basically. Okay, so sort of my final take home, this is really all you need to remember. This is a summary of all the recommendations. Run FastQC before trimming, do your trimming, look again. Always trim paired sequences together because you want to keep the paired sequences in registry and you want to throw away you want to separate out the orphans. Always adapter trim. There's never any value for the primary biological workflows, the bioinformatics workflows of having the adapters on the sequences. At worst, it's going to prevent the sequence. At best, it's going to prevent the sequence from mapping properly. At worst, it'll somehow get incorporated into your assembly, which is something that actually happens quite a bit. Impose a length filter. Here we've used a length filter of 50. If the sequence gets truncated below a length of 50, you throw out the whole sequence. And that's to prevent spurious mapping of the short sequence. For quantification with like RNA-seq, trim lightly. For RNA-seq assembly, trim lightly. For assembly in general, trim lightly. For variant calling, trim stringently, unless you have other reasons, like linkage disequilibrium, patterns of SNPs that you can use to validate. Generally, you don't want to trim your data differently if you're going to eventually combine it. This matters less for things like assembly, but if you're doing variant calling, you're going to have to worry about batch-specific effects from different things, from different trimming parameters. If you use different trimming parameters on your different data sets, the statistical cutoffs that the variant caller is using to determine what's a variant or not will depend. You don't want to have different false positive and false negative rates due to different trimming parameters earlier on in your pipeline, basically. Same for RNA-seq quantitation. There's always this composition bias in RNA-seq. You can ignore that. High coverage RNA-seq is going to have these sequence duplication levels. And then as a final check on these sort of quality score based approaches, take a look at a subset of your reads and try mapping them and just looking at position specific bias within the reads as a sort of a final double check. Now you might think this is all very paranoid or not, I don't know, I can't tell. But something to keep in mind is that once you've done all of this and you're satisfied with it, you don't need to think about it anymore. So you do it, you rest solid on the fact that you've done the necessary controls, in a sense, on your sequencing data, and now you get to worry about the actual rest of things, which is assembling or variant calling and then doing downstream analyses. If you don't do this, then then you could get caught out in a situation where a reviewer notices something. Hopefully you, hopefully not a reviewer, notices something in your paper. And you have to come back and redo your entire analysis because you forgot to do the trimming and FastQC stuff at the very beginning. So there are a couple of references down here. Matt McManus recommends gentle trimming for RNA-seq and has evidence for that. Williams et al. Recommends imposing a length filter. Bondi et al. Has a more complicated message that basically says the first thing you should try is gentle trimming, and then there are some techniques you can use to see whether you should do more stringent trimming. And then this is a paper from 2013 that looks at a variety of common trimming tools and a variety of common downstream analysis approaches and evaluates whether or not you should trim and what tool you should use based on those downstream approaches. I think the recommendations in this hold true in this paper as well, but from this workshop hold true for this paper as well. But it's if you want to read more about what tools are out there and what they're good for, this is maybe a decent place to start. All right, are there any questions at this point? Everybody's like, it's lunchtime, I'm done. We're good here. Are there any questions from the remote folk? Okay, I hope that was mildly entertaining, somewhat informative, and in any case, it's recorded, so I never have to do it again. No, I'm just kidding. So, yeah, we'll just play the YouTube video at the front of the classroom next time. And I have to fix a couple bugs. Actually, if you could make a note about this Kachmer bug, that would be nice. Thank you. So, and please remember, we do have these data therapy sessions. I recommend working with, the takeaway from this is here's some things to think about. Once you have your own real data, that's when to really go back over this and start poking around in your own real data. We're happy to help you with that. So thanks.